to reintroduce myself. It says everything there is to know on there. I want to say this is kind of an old project from almost 10 years ago. So in terms of the tools and some of the ideas, it will look quite obsolete. But I thought it's good to talk about it, especially as the last talk in this week. Everybody is tired because it's a very cool story with some really interesting aspects to it. Also, maybe like the young people might learn some stuff about what we did in the old days from the things I talked about. So the title is a genetic soap opera. So I need to justify that I use this very pretentious term to describe this story. So let's just make sure that we understand what it means to be a soap opera. So if you're watching a soap opera, you expect to see some amazing and unexpected plot turns. You're expecting to have suspense, cliffhangers, and surprises. Very important to have treachery and lying. And one more thing that's obviously always required from a soap opera is to have romance. Now my story doesn't have romance, but why do they put romance into soap operas? One major reason is that they could have models playing the main role, so we are going to okay. The other thing, some very cool sayings that are really coming to life when you hear stories like that. One really nice one that all of you probably know is that nothing in biology makes sense except in the light of evolution, so we should always be thinking about the evolution that shaped the biology that we're seeing. And the other one, no less famous, paranoids can have images. And anyone who's worked with doctors in particular knows they're always very paranoid on who's doing something behind their back and not telling them and lying to them. So we'll see that sometimes paranoids do it anyway. Okay, so I have some very low-level introduction that I'll just breeze right through. So we all know how human genetics work with the inheritance models of the autosomal and non-autosomal pieces of the genome. And we all know, but it's very important to keep in mind this idea that the two main forces that shape our genome are the mutations that happen and the recombinations that basically create different combinations of the mutations by creating haplotypes over generation. So somehow these two processes work. The third one, of course, not mentioned here, but very important, is selection. That some combinations of mutations might have positive or negative effect on the survival of the species. And so these three things together basically create the shape of the genomes that we see. When we talk about functional genetics, then the point is that we have these two separate pieces of information. We have the genotypes and some properties of the organisms that carry these genomes or genotypes, and we're looking for connections between them. But what we are really looking for, so there's two different things we might be looking for, and I'm sure we all know the difference, but let me just read them. One is we're looking for statistical associations between them, but we are really hoping to get at the mechanisms that connect them to biology, right? So, for example, not only the statistical associations, but the actual genetic causes of disease. This is also, this was written years ago, but I think the situation hasn't changed much in the sense that there's lots and lots of studies of connections between genetics and disease. We have a lot of associations, a lot of accumulating knowledge, but our medical understanding of the implications of these lags far behind our ability to find statistical associations. Okay, this is how GWAS works. I'm sure we all know. We're looking for these genetic patterns. The simplest one is just a SNP, a single mutation that are associated with the genotype. And we are hoping to eventually be able to we're hoping to eventually end up with the biological causation, but just finding an association is definitely not yet finding a biological connection. Okay, we are still in the intro, and so of course we understand that what the genome encodes are these proteins and connections between proteins and different functions of proteins, and so we expect that most of the connections we find will be related to the genes and the proteins that come out of them. For example, changing the structure of the protein, and therefore, so changing the sequence is changing the structure, and changing the structure is changing the function, and this is kind of the most basic chain of logic that we would expect. And when we find risk factors that are Mendelian in nature or very strong, even if they are not Mendelian, we can often associate them with exonic mutations, such as BRCA, for example, or genetic diseases that we understand exactly their heritability, the way they are inherited. But typically, and this, since this was written, I think this become much more widely understood, that the weaker associations that we find in complex disease, for example in GWAS, are typically not gene-altering mutations, but they refer to more complex mechanisms such as regulation. So, okay, this is an introduction that basically I was talking about things that most of the people here probably know more than me about. So we might move now to things that hopefully the audience doesn't know a lot about. So, and I also don't. So it would be a good fit. So we'll move to talk about epidemiology a little bit. This is still background. So what we are interested in here is chronic kidney disease. And I'm sure we all know from general knowledge that kidney disease is very common, is very expensive, and is very damaging to people worldwide and in the US in particular. So as of around 2010, or this is actually around 2006, it was claimed that 15% of the US population suffer from chronic kidney disease. And chronic kidney disease is not only being on dialysis, it's also just having some reduced function of the kidney that requires medication, even though you're functioning regularly. So I guess 15% of the US population were on some sort of kidney medication as of 2006, and about 8% DM is tied to diabetes mellitus, and about 2.5% suffered from chronic heart failure. And the very important thing to notice here are these overlaps. Obviously, diabetes is closely associated with kidney failure, although actually the vast majority of kidney failure cases are not related to diabetes. And the other very strong connection is between heart disease and kidney disease. It is well known that people with kidney disease are much more susceptible or have much higher prevalence of heart disease than the general population. But one thing that is extremely interesting and very easy to do in the US is when you compare the ethnic disparity in these diseases. So, for example, many of you don't know, and I didn't know either, that the milder forms of kidney disease are actually more common in European Americans than African Americans. The advanced stages are much more common in African Americans compared to European Americans. And one thing that's really interesting is that if we look at African Americans that have end-stage kidney disease, the prevalence of heart disease is much lower compared to European Americans with end-stage kidney disease. Right? So the conditional probability of heart disease, given you have severe kidney disease, is high in both populations, but is much lower in the African Americans. For this, I have some nice graph. So this is again the conditional probability, right? So the prevalence of heart disease within people with end-stage kidney disease. We see, for example, people that also have diabetes. It's almost 40% in males, in European American males, and about 20% in African American males. And people that don't have diabetes, it's even a bigger gap for males, and I mean similar but less extreme picture for females. Okay, so we have this epidemic that's American-wide, but it has different characteristics in European Americans and African Americans, and very distinct different behaviors. So, so this is where this approach that was very popular, I guess 10, 15 years ago, so admixture mapping is meant to deal with a situation where we have significant racial disparities in some disease, for example, end-stage kidney disease. We know that we have significant disparities both in the prevalence and in the epidemiology of this disease between European Americans and African Americans. And we have a population of recent admixed origin, and we definitely have that, because African Americans are in fact an admixture of African and European ancestry. And the point of admixture mapping is, in that case, we can identify which regions in the genome are basically responsible for the genetic basis of the differences in disease etiology or disease prevalence in these groups. And we can do that by studying the genomes of these admixed individuals, healthy and affected, and in fact we can do it by studying affected genomes only. So this is kind of cool. I'll show you some version of the admixture. So this is called admixture mapping, or mapping admixture by index. So here's the idea. We have a guest appearance by Steve Schiffman, who's not here anymore. So basically what we can do is we take a bunch of African Americans. This example is not the sort of the color scheme is not exactly good for African Americans, because in this example we have admixture with a little bit of orange and a lot of blue, and the orange population is responsible for the risk. So in this case we see that the sick individuals, the cases have in this region in the genome that's responsible for the elevated risk, the cases, most of them have orange, or all of them have at least one orange, whereas the controls don't have orange. And because there's actually a small percentage of orange in the genome, then you know, this is just expressing the fact that there's a small probability of an orange variant in every point in the genome, and this is expressing the fact that to get the disease, you probably need to have orange in that region of the genome. With African ancestry in African Americans, the picture is a bit different, because we know that the major source of the genome is actually African for African Americans and not European. But conceptually, it's similar. We can divide the genome into regions, or somehow estimate in every point in the genome what percentage of the ancestry is from one source versus the other, and look for excess of African ancestry in a region in the genome. And the point is that we could do this with cases only. How can we do it with cases only? We can actually compare every individual to themselves, estimate the overall genome-wide percent of African ancestry for each individual, and look for region in the genome where per individuals and accumulated across all individuals we have excess of African ancestry. So we could do this with cases only. We don't actually need controls. And there's one specific algorithm that does it. It's called admixture mapping. It's hitting all the keywords for methods that we can think about. It's a hidden Markov model to infer what percentage of the ancestry is African at every point in the genome. So we know that this is a continuous process, right? If I have African ancestry in one point in the genome, I'm highly likely to have African ancestry close by. Why? Because the stretches of ancestry in my genome are actually determined by the recombination, right? If it's recent admixture, then each chromosome might be might have been broken in 10 or 15 places by accumulating recombination since the admixture event. And so I would have like 10 or 15 pieces to each genome. Within each piece, I would have a fixed origin, European versus African, right? So basically, the recent admixture means that relatively long stretches of the genome would be from the same continental origin. And within the HMM, we have an MCMC, so that now relates to my talk from last week. And why did people really like admixture mapping circa 2008, 9, 10? So one important point is that it requires much less data than GWAS to identify the disease loci, because we have a lot of power because we have long stretches in the genome. So this really allows us to infer with high power, with a relatively small number of samples, which regions in the genome are. So this is not fine mapping like GWAS, or not pinpointing like GWAS, but this is identifying regions in the genome. So that's the advantage of GWAS. We have more power with less data. The advantage over traditional linkage is that it does not require Mendelian inheritance, so it doesn't have to be the same mutation, right? It's more similar to the overall gene load test that people are doing now, because it's identifying regions that might be a gene or several genes wide. So this was, and one very important thing is that to know is that at that point in time, people were not very rich, were not basically doing sequencing. So sequencing things as a way of solving problems was much less relevant than it is today. So we were limited to doing SNP chips, and it would be much cheaper and more realistic to do, say, 1,000 SNPs across the genome, which is what you need for for admixture mapping, than do 1 million. I mean, the cost would be disproportionate. So that's why admixture mapping was really popular. And this end-stage kidney disease difference between European Americans and African Americans was really a good candidate for this kind of study. So people went and did this study. And in 2008, in Nature Genetics, 2 independent studies using, supposedly independent, I mean, they were using different data for sure, but they reached exactly the same conclusion. And the conclusion is trying to find the region in the genome where the genetic basis of this difference lies, right? So, and they both found the same position in the genome, same region in the genome, original chromosome 22, and they also both had a suspected gene, supposedly independently, and the gene name was MYH9. So the conclusion of both of these studies, and I just want to introduce 2 of the stars of our talk, Jeff Kopp and Cheryl Winkler from the NIH. So these are the big shots in kidney disease in the NIH then, and I think possibly also today. And so they were the first and last author of one of the papers. The other paper, I think, was from John Hopkins. Okay, so both of these groups.
Dates on those 2 papers? I mean, how close were they in time? They're both.
Oh, they appeared at the same time. I guess they were submitted almost simultaneously. I'm sure they knew about each other, and they submitted. I don't remember the history. Maybe they submitted together, or maybe they submitted separately, and the journal said, we have these 2 papers, we should give both of them a chance, so publish both of them at once. They actually appeared in the same issue of Nature. I mean, it's very redundant to have 2 of them, but that's what they did. Okay, it's also, we can talk about the politics behind it. So why MYH9? So first of all, there was some known connections between MYH9 and kidney disease. So these people, when they found that the peak of the admixture mapping was very close, was in the region of MYH9, they were really happy because they knew the name. As kidney doctors, they knew the name of MYH9 from some other phenotypes, but that are related to the kidney. for example, this second point, the term mutations in MYH9 had been shown to cause familial clusters. So in linkage studies, they were actually able to assert the real connection between MYH9 and kidney disease. So obviously it's a good candidate. So these were kind of the biological reason, and the statistical reason was that, so we are saying that what we're doing is we are, by the HMM, by the hidden marker model, in fact, we are inferring at every point in the genome how much of the percentage of the ancestry is African versus European. And this is a continuous function because the HMM is sort of continuously, smoothly changing. And we can look where in the genome is the biggest difference between sort of the genome-wide African ancestry and the local African ancestry. And the first group, the NIH group, actually got that the peak is right on MYH9. So that was actually where the highest peak in the genome. So they had one SNP in MYH9 in the panel, and that was the point where the highest signal was observed in the HMM. And they also, I'm not sure how to do for time. These links lead to figures and tables from the paper. The other thing is they did tests like we do in GWAS for some genotype mutations in MYH9, and they found a strong statistical association between these mutations, and which, if it didn't happen, it would be very surprising given that the mold peak was on MYH9. So you could say that they confirmed the statistical association by doing genotyping of SNPs and getting a strong, very strong statistical signal about association between MYH9 and end-stage kidney disease in African Americans. Okay, so it's also nice that they looked in other genes within that region. So that region was centered at MYH9 for one paper. For the other, it was actually centered a bit away from MYH9. So they said, I mean, because the signal we find is not super local, we can look at 5 genes to the left and 5 genes to the right and select some SNPs from those, genotype them as well, and look at the strength of the statistical association. And basically, to some extent, they found that whatever statistical association existed is explained by LD between these mutations and between these SNPs and the MYH9 SNPs. So all the signal pointed to the fact that we should be concentrating on MYH9, right? And the people leading this area are people who really care about the medical understanding and medical implications. And so, and this is a very important phenotype with a very striking finding in an interesting gene. So basically, everybody in the world was working on trying to understand this connection further. This is over about a year and a half from the publication of the original paper. This is just a sample of hundreds of papers that appeared. For example, a focus on the MYH9 susceptibility gene. This is related to HIV-associated nephropathy. So people with HIV are much more likely to develop end-stage kidney disease, and people with HIV and this MYH type thing are like 100% probability or many tens of percent probability to develop end-stage kidney disease. And the point is that I became part of a group that was also working in this area at that time. So this was actually a group in Technion led by Karl Skoretsky, who's a nephrologist and a scientist, and Doron Behar, who's a well-known geneticist. So these are people from Technion, and I was working with them. And we also collaborated with these two people, Kopp and Winkler from the NIH. And what we were doing is we were looking for interesting things that we can say about MYH9 associated kidney disease, trying to have new statistical perspective or new biological perspective. So this specific paper is about a not very interesting fact that the same effect that we see in African American replicates in Hispanic Americans, and Hispanic Americans have a much lower but non-0% of African ancestry. So you can actually find the same mutations in MYH9 in lower prevalence, and see that they are actually associated with end-stage kidney disease in Hispanics as well as African Americans. Not very interesting and very expected. I should mention that we had our data from this second to last guy, Walter Wasser. So the cases are just a bunch of cases from one clinic in New York City, just from one dialysis clinic in New York City. He just brought his patients with him, and we had some crappy control set to go with it. And so we were collaborating with Kopp and Winkler in having these weekly or bi-weekly phone calls to talk about what interesting things we can do, both in terms of data analysis, statistical analysis, looking for new frontiers in this. And this is, we're now in February 2010. April 2010, there was, to demonstrate how important this thing was, they had a special conference at the NIH about MYH9 in kidney disease, clinical and public health implications. And of course, this is in NIH. Two of the stars of this are our friend Kopp and Winkler, and we're talking about April 2010. Okay, so now we stand in April 2010. We're having conferences about MYH9 and kidney disease and what can we do about it. And what do we have as of April 2010? So we have many independent replications of these original statistical findings. For example, our Hispanics paper, which is just replicating the same thing in a slightly different context. So we know it's real. We know the statistical association is real. They did more and more fine mapping on MYH9 and found additional strongly statistically associated variants in MYH9. So we have one way of saying it, we have multiple independent signals that do not explain each other within MYH9. There's no progress on how the biological mechanism makes sense, and none of these mutations are actually mutations that are supposed to have a major functional effect. So I think they were all intronic, or might have been a couple of synonymous mutations in exonic regions of MYH9. So now we need to go back and ask ourselves, given that we have a strong statistical signal, but we really have no progress on what the biology is, should we really concentrate all our efforts on MYH9? And I was the one actually pushing it, that we should try to look for other genes and other mutations, and we had a phone call with the NIH people, and we told them maybe we should be looking in other places. That was the last phone call we had with them. We'll see why in a little while. But we didn't have good data. We didn't have a lot of money. And so the point was, let's look in the public databases. Let's look in the literature and find things that we think might be interesting. Then we can invest money in genotyping them and on the case control data and examining them further. So, for example, other mutations outside MYH9 that could conceivably explain the association within MYH9, given, for example, LD patterns. And the main source as of April 2010 was HapMap. I hope you all know what HapMap is. So HapMap was supposedly a map of human genetic diversity. Before we had sequences for different populations, this was supposed to be a pretty exhaustive map of the common variants in different populations in the world. So if you try to read and understand what HapMap claims to have, then it's supposed to have almost all common variants in different populations that have been studied. Okay, so for example, there's a large number of Yorubans, West African samples that are part of the sort of the original discovery samples of HapMap. So supposedly we would expect that most of the common variants in West Africa would also be present in HapMap. But my search of HapMap and other resources found no promising candidates. Yeah.
What was the
in MYH9? High, like 30-40% in say YRI in Africans, and maybe 10-15% in European. there was major frequency difference, of course, because we're looking for something that the allele responsible for it should have major frequency difference between the populations, but it was pretty common in Europeans as well. Okay? And this should be a hint, right? Or it was not super rare in Europeans as well. This should be a hint because this, I haven't really talked about it, but one, and we didn't understand it until later, one major difference between end-stage kidney disease, non-diabetic end-stage kidney disease in African Americans and Africans against Europeans, is that you have non-negligible several percent or like 1% in the population prevalence of end-stage kidney disease that's very early onset in African Americans. Like people by age 40, they need a kidney transplant, and this basically has zero prevalence in Europeans and European Americans. And so this tells you, this should be a hint that we didn't really think all the way through then, that what you're looking for probably has zero prevalence in Europeans. Okay? So, so at that point, something that's well established today was only in its pilot stage, which is the 1,000 genome. So 1,000 genome is a way to not depend on HapMap for finding variants, to be able to find your own variants. So around April or March, the first pilot data of the March, April 2010, the first pilot data of the 1,000 genome project came out, and it was 60 European genomes and 59 West African genomes. And once you start to look in there, you suddenly see completely different things. Okay, so we said let's focus on the most smoking gun that we can find, which is non-synonymous coding mutations that are in genes close to MYH9 and that they have major differences in allele frequency between Europeans and Africans. Basically, they have to be relatively common in Africans and missing completely or almost missing completely in Europeans. And once you look at the genomes, you start finding many of these. So none of them are in HapMap. These are 8 mutations in neighboring genes that are non-synonymous, prevalence at least 10% in the West African genomes that we had, in the 118 West African chromosomes that we had, and prevalence 0 or close to 0 in Europe. So now we could go and the gene, we had 8, we are going to genotype all of them, but the gene, the answer is going to be in this gene APOL1, which is actually the next gene over. This is MYH9, this is APOL1, so it's actually the next gene over from MYH9. And 2 of these of the 8 we found were in this next gene over, and in fact it's not 2 but 1 because they are in perfect LD. They are perfectly co-inherited. In that data they were in perfect LD. In bigger data sets they are in almost perfect LD. So it's like one haplotype. It's only about 20 kilobase from the best MYH9. So the best MYH9 mutation is not one mutation but several mutations, but they're actually on the side close to the APOL1 gene, and they are amino acid changing. So this is very exciting. Now we can do the statistics. So now our small preppy samples that includes like 100 cases and a bunch of controls, we can do the statistics and see that the strongest association signal we find by far are in these APOL1. So we see that they are not in perfect LD, they are almost perfect LD, but the strongest, the smallest p-values, the strongest signal we find is on these APOL1 mutations. This is compared to the MYH9 best mutations that we have. Okay, and we can also do an exercise like put this in a model and then see that these are no longer significant, right? So see that this signal is actually explained by this signal. So we have strong statistical evidence that APOL1 is in fact the one responsible. But does APOL1 make sense biologically? And the answer is yes, because there was already knowledge in the literature that APOL1 is associated or related to Trypanosomiasis, which is African sleeping sickness. Okay, so African sleeping sickness is traditionally or evolutionarily a big problem in Africa. and humans are naturally immune or developed immune against most of the parasite species. So once you get infected, if you're not immune, you basically die from this not very nice disease. So this is a parasite, and the human serum is able to kill most of the species of this parasite, but 2 species are still responsible for disease in human. And in 2009, they showed that this protein, A2L1, if you change it in some ways, it's able to also kill these 2 species. That basically gives protection. You can give the body protection against these additional species by changes in A2L1. So this was supposed to show that West Africa is actually traditionally and still to this day a place where African sleeping sickness is a major concern. This is the origin of most of the genetic material of African Americans. So one thing we did is test these study these mutations and figure out that they are related to the ability of the APOL1 to kill this parasite. So basically, some prediction mechanism says that they are related to the change in function that allows to kill the parasite. So now we are in mid-May 2010. We are the provincial group in Technion and Tel Aviv University in Israel. We've been disconnected from the NIH people who don't return calls anymore. And what we have something that we believe is a major finding. We have a theory about an evolutionary connection to African sleeping sickness. We have very lousy data on the statistical side, and we have 0 data on the biological connection to the African sleeping sickness. And a very strong sense that we are being threatened by these large, well-funded and data-rich groups who are probably in the same place or ahead of us. So what could we do? We could look for more data, more collaborators. We could try to find a way of doing the biology in the lab, or just try to publish what we have. So we decided to try to publish what we had, and ambitiously sent it to Nature Genetics. And they, as expected, replied, this is very nice, but it's not really solid. We need better data, and especially we need something about the biology if you want this to be Nature Genetics worthy. So we contacted another well-known guy, trying to get more data and other things from him, and he said explicitly that he was part of a group that is making major progress on this general story, and he cannot work with it. Okay, so the conclusion, and then there was one more thing that I wanted to show here, but the video is no longer online. There was a 1-hour talk by this guy, Jeff Koch from the NIH, about MYH9 at Wake Forest. He was giving a highly attended talk about MYH9, and the whole 1-hour talk was about MYH9. This is late May 2010. And in the answering question part, people asked him, what about connection to disease? And he said, well, we should look at different diseases. And he said the word Trypanosomiasis, which is African sleeping sickness. And at that point, my doctoral collaborators from the Technion, who I was sure generally always think they are being chased by it, said if he says this word, for sure they know it's APOL1, and for sure they are going to publish it soon. So because of this word, we have to publish immediately, no matter what. Okay, so that was the conclusion from this one where the mounting evidence was that we are paranoids who are being chased. So, okay, actually this is a very important slide. So this is our paper, and there are several things we can see here. One, where it was published, this pretty obscure journal, Human Genetics. But what's the benefit of working with an obscure journal and publishing something important in an obscure journal? We submitted the paper on 7 June, and it appeared online on 16 July. Okay, so the point was, we're going to give you this paper, but you have to publish it basically immediately. Review it within 2 weeks and publish it within a month. And now the question is, were we really being chased? And the answer is yes. So our paper appeared online on 16 July. This paper appeared online on 15 July in Science. And it says association of trypanolytic A2L1 variants with kidney disease in African Americans. And when we look in the author list, we find some nice surprises, such as our friends Winkler and Kopp and our other friend Barry Friedman. And we also see that this paper was originally submitted on 4 March 2010. In April 2010, there was a big conference in the NIH about the other gene, led by these people from the NIH, who are co-author on the paper submitted in March, saying that it's actually a different gene that's responsible for it. Right? So they already had submitted a paper saying it's a different gene, and they had a conference on the wrong gene. And this is part also of the cultural phenomenon or big problem maybe in the science in general, medicine in particular, that before something is published, it's not considered known. Right? So even if you know something, you hide this knowledge and you have a big conference and you go to Wake Forest and you give talks about something you already know to be wrong because you haven't yet published that it's wrong. Okay? So, so one question we might ask ourselves, except the gossip and the doubts that we have about the scientific method, was there the justification that this paper appeared in Science and our paper appeared in a third-rate journal, and actually there was. So they were able to actually solve all these things that we would have wanted to solve but couldn't because of our data limitations. Turns out also some intellectual limitations, and we did not have labs to see what these things do to the trypanosomiasis parasite. So one very nice thing that they did is they found an additional mutation, which is a deletion, which we would have been able to find if we knew how to look for it in 1,000 genome, more rare mutation. But when you combine this other mutation with the mutation we also found, you actually get pure recessive mode of inheritance. We found an additive mode of inheritance because we didn't have both mutations. When you include both of them, so you need a copy of one of them, of at least one of them on each of the chromosomes, and then you have the increased risk, and it's purely recessive. I'll show you the proof of that in a minute. And the other thing they did, which is amazing, they actually proved in the lab that this mutation, these two mutations, give protection against that. So the theory we had as a theory, they proved it in the lab. 5 minutes? Okay. So this is, for example, proof of this is proof of the recessive mode of inheritance. This is these go up top. There are the mutations we found, and we see very strong signal just doing a simple test of association. So this is low 10 p-value. We see weak. Once we remove those, we see a remaining weak signal up top for the 6 base pair deletion. And once we remove both of them, so we include them as explanatory variables in our logistic regression, we see no signal left for MYH9 mutations or any other mutations. Okay? So we've proved that by using these two mutations, in particular in a recessive mode of inheritance model, we explain all the signal, all the association signal in the region. And this is the proof that these things kill parasites. This is the nicest thing to look at. So actually, one nice thing is that they kill parasites in a dominant mode, inheritance mode. So you need just one copy to kill the parasite. You need 2 copies to get sick. And this is what happens. This is a happy parasite, and these 2 are dead parasites where the thing connected to them and killed them. Okay, so these are actually pictures they were able to generate in the lab, and these are graphs showing that not all of them kill with the same effectiveness, but generally demonstrating the ability of these variants to kill the parasite. Okay, so I think we understand what the emerging theory is, that these mutations appeared in West Africa because they were protective against trypanosomiasis. Some variants, they went through this selective sweep. And at that point in time in Africa, probably kidney disease was less of a major threat to survival than African sleeping sickness, so they were actually positively selected in West Africa. And obviously this moved with the gene flow from Africa to the US. In the US, these African Americans were not exposed to trypanosomiasis. On the other hand, they have increased lifespan and maybe some environmental factors that like HIV, which is not exactly an environmental factor, and then the kidney disease risk of this emerged as a major issue. This is just demonstrating the selective sweep. I don't think it's clear. So actually, people have been working. So this is, we're talking the second part of, or middle of 2010. Since then, many people have been working on the really there's one major piece of the puzzle that remains. What's the mechanism of getting kidney disease with this? So what's the actual connection of APOL1 to kidney disease? We know the connection to trypanosomiasis, but what's the connection to kidney disease? This slide is out of date, but when I try to look today, I don't think they have made any major progress on understanding the biology, the connection between the gene in particular and these non-synonymous mutations in this gene in general and the non-synonymous mutations in particular to kidney disease. But there's ways they are using it. For example, they are screening potential kidney donors of these mutations, and they do not take kidney donations from people with the risk genotype. This is one way this is actually medically used. Okay, thanks.